I'd like to discuss first some considerations for plant cell culture. The first of these relates to choice of explant. We very rarely put a whole plant into culture, but rather choose a part of the plant which has some desirable properties. For reasons that you'll see very shortly, it's helpful if the explant is easily sterilizable. It's helpful if it's juvenile, and it's helpful if it's responsive to culture. So this means that the ideal explants are things like shoot tips, axillary buds and seeds. Sometimes we can use hypocotyl from germinated seed, sometimes we use leaves. When we look at an explant, for example an axillary bud, so in any one particular plant we'd normally chop out an explant out of the plant. Everywhere that a leaf comes off the stem of a plant, you have a bud in the axil of the leaf, and this bud, in a mature plant, it will grow out and give you a branch. In a juvenile plant it will grow out and give you a new shoot, and this is the basis of commercial micropropagation, or at least 90% of commercial micropropagation. Or we could go for something like the shoot tip, which has very many leaf initials on it with an axillary bud in the axil of each leaf initial. But whatever explant we take, we're chopping it out from the rest of the plant. So rather than taking the whole plant, which has got the shoot tip, it's got the leaves, it's got the roots in it, it's got nodes, let's say we're just taking this nodal cutting here with an axillary bud in the axil of the leaf. We have to replace everything that the rest of the plant would supply. So from the shoot tip you're going to get things like plant hormones like auxins and gibberellins. From the leaves you're going to get a carbon source in the form of sucrose and hormones like gibberellic acids. From the roots you're going to get water, vitamins, mineral salts and another plant hormone, cytokinin. Now all of these have to be resupplied to your explant when you've removed the sources in the explanting. So we need to have a medium formulation, and the medium constituents will have an inorganic salt formulation that will be supplied by the roots, a source of carbohydrate that will be supplied by the leaves, vitamins that will be supplied mainly by the roots, water, a variety of plant hormones depending on what we want to do with the explant. Typically we'd have some solidifying agents. It is possible, for example, to chop a piece of stem out, put it into a test tube or a boiling tube with some liquid and grow it in liquid media, or to put the explant in a shake flask and grow it up in a shake flask in liquid medium. And this can be a very efficient method for example, micropropagation. But for some explants we need to maintain the polarity in the plant. So normally we'd supply some kind of solidifying agent like agar to make the medium easier to handle and to maintain polarity in the plant. There are also a range of undefined supplements which we can add to plant cell cultures.